Hello, my name is Jacob. I am a peer advisor with Project RAISE, and today I'm going to give you an overview of poster presentations. In this overview, we're going to cover four main topics briefly. What is a poster? Preparing it, smart posters, and some general tips to help you get started. So what is a poster? Well, it's a communication aid that provides a summary of the current state of a project using a combination of figures, tables, and text. Like a PowerPoint, it's used as a reference when communicating with your diverse audience, which can consist of anybody from professors and professionals that you want to work with to students passing by. And because of this, it needs to be formatted in a visually easy to understand format. So that way, if you need to step away, anybody who walks up to your poster can read it and understand what questions you asked, how you approach those questions, and what were the results of your approach. Now that we have a basic understanding of what a poster is, let's figure out how to prepare the actual poster, starting with who your intended audience is. Is this going to be a giant interdisciplinary conference, or is this going to be a more local conference with professionals specifically from your field of study? This information will help inform your discussion with your principal investigator on the aspects of your project to highlight, what data to present, and how to present it. Next, you want to begin working one section at a time, starting with your most specific information. In general, this means that you're going to be working with your results and your materials and methods section first, and then broadening out to your discussion and introduction. As you add each successive section, figure, table, any visualization, you're going to want to look back, edit, and reformat your poster as needed. After you are satisfied with your poster, you're going to want to practice. And you're going to want to practice a fair amount of times leading up to the conference. So that way, when it comes time for you to present, you can confidently stand by your poster and wait for the next audience to come. So let's start with the results section, because as I mentioned, we want to go from the most specific and relevant information and broaden out. And in this case, that broadening out goes to the discussion section. For the results, you want to highlight the most relevant findings of your study. And this can be achieved through the use of figures. Now, figures can refer to graphs, photos, and even web-pulled images. And they should generally be formatted so that they appear in chronological order of when they will be generally referenced. They also all need to have specific figure captions that describe the who, what, where, when, and typically how of the figures that are being depicted. Now, specifically for graphs, you need to make sure that your axes are labeled with units, that your data sets are distinguished by both symbol and in the legend. Typically for graphs, you also want to remove the grid lines so that way your data can be highlighted in the figure. For photos and microscopic images, you typically want to put in a scale bar or indicate in the figure caption the magnification. If you present more than one figure within the same figure section, you can also label them A, B, or 1 and 2. Finally, for web-pulled images, you typically want to cite your source, even if it's a watermarked web address at the bottom of the photo. All this to say, figures can be a very versatile and useful tool in order to present your relevant results. Also for your results, you have the option to present your data in the form of tables. Note that if you do so, do not present any data that is oriented in either the text or in any graph features. But for tables, you generally want to have these components shown here in this example. You have your descriptive title caption, you have your categorical variables, you have labels of different treatment groups and studies. In addition to labeling your replicates, you have the relevant data points that you want to highlight, and you have footnotes to indicate any exceptions or abnormal treatment groups that occurred during the study. Tables can be just as effective as figures in presenting your data, so you really need to think about which one you really want to use in your specific study for each individual part. The next section that you will want to focus on are the materials and methods. This is where you get to describe to your reader how you got the results that you did, which is important because you want to ensure that your experiment is replicable. Now, these sections can vary from poster to poster, experiment to experiment, but in general, you want to make sure that your text is limited and concise by omitting any jargon and you substituting it with umbrella methods. For example, the example on the left indicates umbrella methods like DNA extraction, PCR reactions, and plasmid extraction. You also have the option of using the headings and subheadings to your advantage, like the section on the example on your right. This student indicates that this section is specifically dealing with their morphology, materials, and methods. In general, you also want to make sure that you reserve part of your materials and methods section for data manipulation, because how you altered the data is sometimes just as important as how you collected it. Lastly, as you can notice from both examples, the use of figures is highly encouraged because they can be used as a reference for the reader and your audience members on how you specifically conducted certain methods. For example, on the example on the right, the student can point to the mouth at A and indicate how they took the measurements for their calculations. If taking photographs of your methods is not reasonable or applicable, you could also use clip art or different programs to construct your figures and flowcharts. One free program that is fairly easy to use is called BioRender, that can be used to create very effective material and methods flowcharts. In general, in these charts and in your text, make sure that you refrain from just simply listing out your materials and just introduce them as you use them throughout the text. The next section that you're going to want to work on is the discussion section. In this section, like the results section, you're going to want to start narrow and broaden out to the bigger picture. This means that you will want to start with your expectations versus reality. And in doing so, you will frame your results in light of the hypothesis or the objectives that you introduced in the introduction. Whether or not the results met your expectations, you will need to provide scientific basis from the primary literature to support what you actually found. This is also a section where you may indicate where you think things may have gone wrong. Although nobody likes to present bad data, you still need to reflect on your own experiment. And in fact, you can use that to establish the future directions of your study. Lastly, in this section, you also want to establish how your study fits in the overall picture, the global picture. What do your results say about the future of your field? And how can future studies build on top of what you have done in this one? In the example provided, we see a similar pattern of starting somewhat narrow and broadening out to the bigger picture from the first to the last bullet points. The first bullet point provides a specific reference to the data and the expectations that may have been referenced in the introduction of the hypothesis. The last bullet point frames this whole study in how it fits into the bigger picture of restoration efforts. One of the last sections that you will want to work on is the introduction. This is where you use your knowledge of the study subject and the problem through your primary literature searches to really set the stage for your entire study. In this section, you will want to make sure that the audience has enough information to be able to follow along and logically come to the research question of whatever your study is looking into. The question differs from the hypothesis by the hypothesis essentially being the proposed answer to the question. The hypothesis needs to be supported by both observation and previous studies work. So this is one of the sections where you will really need to incorporate your primary literature searches. The hypothesis differs from the scientific objectives by essentially being broken down into those objectives. The objectives are used to either validate or invalidate the hypothesis. Think of the objectives as a short-term goal to achieve the long-term goal hypothesis of the study. In the example to our right, the student starts broad in this introduction by introducing us to the brewing industry in California and successfully narrows down the information to the main problem of gynostine affecting the reproductive physiology of developing organisms. This allows the audience to infer that the question is, what are the morphological effects of gynostine on a certain organism? In this case, it's a zebrafish. Their proposed answer to this is that the zebrafish will essentially experience smaller body weight, length, and more developed ovaries at various doses. So this hypothesis can further be broken down into the various objectives, inferred to be model the concentrations of gynostine found at a brewing outfall site, or even rear zebrafish to varying concentrations of the chemical to understand the relationship between the concentration and the morphological structures. One of the last things that you need to work on are your title, your abstract, your acknowledgements, and your works cited. For your title, you want to make sure that it's large and easy to read. So that way somebody who is passing by about 5 to 6 feet away can easily interpret what you have on top of your poster. This is also the section where you include the logos of your associated institutions and programs. And your title does need to be smaller than 20 words, not including your authors, and below that, the institutions that they are a part of. Next is the abstract. This is a paragraph of about 250 words that provides a summary of your study, introduction, methods, results, and your discussion. This is not always attached onto the poster, but is typically used with the title in conference agendas, so that way readers and the audience members can look ahead of time of posters that they may want to visit. The acknowledgements section is for any contributors to your project. So this includes funding sources, individuals or institutions that loaned you equipment or techniques, or even maybe a study organism, specialists who perhaps operate microscopes or specialty equipment, and even assistants. So perhaps your lab mates who helped stay with you and pipetted certain samples. Make sure that this section does not need to be as large of font size as the other sections. So that can take that into account when formatting and resizing and repositioning your poster. And finally, the works cited. Similar to the acknowledgements, it can be reduced font size, but this is where you list in order the references that you received certain pieces of information in the order that you refer to them. After writing and rewriting all your sections, titles, figures, and tables, you come out with a finished product like this. This is a finished poster presentation. Notice the title is descriptive. It includes the authors, the institutions, and the programs that they are a part of. The student decided to include their abstract, whether it was due to the conference or by convention of their field. They included a goal, or essentially an objective section, along with their introduction. They decided to put the main focus on their results and leave their conclusion as a side abbreviated section. And as you can see, they did still include their acknowledgement. No two posters will ever look the same, so you need to really discuss with your principal investigator how best to represent your data for your study. Now, in recent years, there have been the development of things called smart posters. These are posters with built-in technology like QR codes that allows the audience to interact using their smartphone. These QR codes typically lead to a lab website where you or your principal investigator can publish extra results, methods, and other information relevant to your study. Think of this as the supplemental information in primary literature articles. Lastly, we want to leave you off with some general tips in preparing for your poster presentation. First off, start early. As you can see, there's a lot that goes into a poster presentation. So making sure that you have enough time to write and rewrite and create your figures will really give you a reduced stress for the conference ahead and allow you to focus on preparing networking opportunities or even just your presentation, what you're going to say. Next, you're going to want to stay in contact with your principal investigator. Remember, they are here to help you. They are experts in your field, and they know how best to represent your study and your data. Discuss with them and come up with your own ideas and present it to them and see what they think. Next, make sure you research the guidelines of your conference. If they require an abstract, you need to know that beforehand so that way you can visually lay out your poster in a way that makes sense. Some posters are required to fit in specific dimensions, and some posters are even required to have QR codes. So really do some research and look ahead as to what is required of your poster at your conference. Next, as we've emphasized all throughout this presentation, make sure that you start with your most specific information first: the methods, the results, and the introduction and the discussion. This way you ensure that you put focus on the items and the sections that really matter to your study. In preparing for your actual presentation, have ready 3 intensities of presentations. typically about 30-second presentation, a 2-minute presentation, and a 5-minute presentation. This represents your preparedness to handle any intensity of audience that you may have. So a 30-second is perhaps a passerby that may be waiting for another poster, someone who you can maybe tell may not be well versed in your field. Whereas a 5-minute presentation is perhaps someone who really is invested in your field, and you can tell that they actually have some knowledge or background in this area. Or perhaps you want to establish this specific network contact. Even if you prepare these 3 presentations, also be prepared to improvise and start from any section on your poster. Remember, you are the expert in your field, so picking up in the methods, if they have a specific question for one of your methods, should be no problem. And lastly, go ahead and look for external resources to help guide your writing process. There are many books and online articles and PDFs that are available to students, a lot of them for free. So really look to see what is available to you. LabMates are able to help provide maybe some insight or even templates of previous posters. That way you don't have to start from scratch. I hope you found the information useful in this presentation, and I invite you to connect with us at CSUF Project Raise on our Instagram and Facebook. You can email questions to raise@fullerton.edu or visit our website www.fullerton.edu/projectraise/. Thank you.